So when we look at the context of the endocrine system, our outcomes really that we're looking at specific to exercise are things catabolic, are they breaking things down, or are things anabolic, are they breaking things up? And we were introduced to this anabolic and catabolic states when we were looking at molecules for energy production, right? So we looked at when we eat something, do we break it down to its smallest components, or when we then want to store things, like if we want to store substrate for later ATP use, are we building those things up? And so we looked at the net sum of anabolic and catabolic reactions. That's how we came up with our metabolic output. And we looked at this from food is our stored form of energy. We eat that. We have catabolic processes that break that down into smaller components, so like amino acids, and then we have anabolic processes that, and then we usually stop there. We stopped there in our conversation last unit because we were using that catabolic to break down substrate to produce ATP. Well, what we're going to talk about now is the next part here where, yeah, we ate this food, but we're at rest. We don't need all this excess energy. How can we store this for when we actually need it? And so then we get into the anabolic aspect. And so we had this chart before where we had food intake at the top, and you have your three primary macronutrients that are energy yielding, your protein, carbohydrates, and your fats. And your body breaks down the proteins down to their smallest component, which are amino acids. Complex carbohydrates are always broken down to their smallest components, which are single glucose molecules, and triglycerides or fat are broken down into glycerol and fatty acids. And then your body determines what it wants to do with it. If it needs it right there and then, it's going to convert that into energy, or if it doesn't need to, it's going to store that in protein synthesis, and then anything left over, it will then convert to excess fuel, but it'll always favor protein synthesis first. Same thing with fat. When you eat fat, the body will use what it needs first, and then anything left over will be broken down and stored. You notice here that glucose has no other function except for energy production. That's it. It will store glycogen as it needs for excess energy, like it's in its battery, in the liver and in the muscles, and then anything left over, it's going to be converted to fat and stored, right? So then when we need it, so you can see the catabolic reactions here. This is breaking things down, and then you have a few of these anabolic reactions where it's resynthesizing things back into the things that it needs. So when we look at the tissue storage now, so where things can go, you have different cellular responses that occur to stress. So when we look at the same approach with tissues, taking substances and storing it as energy, like I said, recall carbohydrates the only fuel. So what we're really looking at is protein and fat storage, and any excess protein and any excess carbohydrate ends up becoming fat anyway. So we really want to focus on how fat is stored, and we really want to know and appreciate how carbohydrate is stored. But when we look at the storage sites, you have your normal cellular structure here at the top, upper left-hand corner, and when you look at cellular changes, you have one of these 5 things happening. You can either get hypertrophy, so you get increased size of the cell, so you're not changing the number of the cell. You can get atrophy, so that would be happening in catabolic aspects. So the number of cells doesn't change, but the cells get smaller. You can get hyperplasia, which is the increased in number of cells. So you're going from 5 cells to 7 cells. It's like a splitting. And then the other 2 at the bottom there, those are typically in high stress environments, cancer, or really early on in life with differentiation. So you get metaplasia, which is a conversion of cell type, where you get dysplasia, which is usually some kind of cancerous or dysfunction. So what we're going to talk about in terms of cellular responses are these 3 things here: hypertrophy, hyperplasia, and atrophy. And just to kind of disclose, unless you're talking about severe situations, you're talking about hypertrophy or atrophy in our aspects. But the question is are you talking about fat changes, or you're talking about muscle changes, or both? And this is where we get that conversation where I kind of talked about earlier, is that people always think anabolic. Oh yeah, that's good, that's great, because we associate anabolic steroids, and anabolic steroids are associated with increased muscle. But if something was anabolic to, say, fat tissue, well, that's something you want to stay away from, right? Or if something was catabolic, that was breaking things down, if it's talking about muscle tissue, hey, you might want to stay away from that. But with fat tissue, tell me more. I'm interested in that. So we really have to look at, when we look at hormones, and we have to understand the context at which these anabolic and catabolic environments are in. So if we're looking at fat tissue, anything that's anabolic would be, hey, let's put the brakes on that. We're not interested in that. But for catabolic, we want to go full steam ahead. And same thing for lean body mass or muscle tissue. Things that are anabolic, yes, now we can use our traditional perspective, that might be good. Or things that are catabolic, we want to stay away from. And hormones aren't easily say that, hey, this is an anabolic hormone, this is a catabolic hormone. Hormones have different effects on different aspects. So you have some hormones that follow this grid up here that, like growth hormone would be anabolic to muscle tissue and catabolic to fat tissue. So that's a great thing. Things like glucagon, that is catabolic to fat tissue, so we say it's a good thing, but it's also catabolic to muscle tissue as well, so not so good. And we look at things like insulin, it's the most anabolic hormone to lean body mass. It's also the most anabolic hormone to fat mass too. So these aren't hormones we want to like maximize. These are hormones that we're going to want to optimize. So we'll look and explore at the different hormones that are available, but this is the first part of that, is really appreciating, are we talking about lean body mass or fat mass when we're talking about the anabolic or catabolic state? So on the right here you see the two cellular structures. So we look at the bottom right here, and that's the cellular structure of a muscle. Remember, the muscle fiber is like a long human hair. It's visible with the naked eye, and what's unique about muscle fibers is that they have multi-nuclei. So they have different nuclear, they have more than one nucleus. At the top there you see a fat cell, and the fat cell is an interesting aspect, but you can see it has that rough structure, it has a nucleus, but it has that fat reservoir. And on the left there you can see the two aspects. Are we talking about hypertrophy or hyperplasia? So when we look at muscle fibers, the evidence is strong that muscle hypertrophy is the primary aspect for muscle growth, and the way that hypertrophy happens is that you get signaling in the nucleus. The nucleus then increases protein synthesis, and you get more protein stored within this aspect. You tend to get hypertrophy where the cells themselves get bigger, but you don't tend to get hyperplasia where you get multiple cells. Now you might get, and we'll talk about this later, you'll get more protein filaments in here, but you're never going to, you don't really generally change the muscle fibers, the number of those fibers. There's some evidence of that happening, but that's usually in like injury or damage where you've worked out so flippin' hard that the body's basically producing scar tissue through this hyperplasia type production. Fat cells, same thing. It's generally tended that they go through hypertrophy. So when you lose fat or gain fat, you're not necessarily adding or reducing the amount of cells. You're just changing the size. But that's only true when you're talking about lean or normal weight individuals. When you're talking about obese, and not like super fat, clinically obese, but just like this baseline obesity, like 30% or 30 BMI or higher, there is evidence, because there's the big debate with stem cells. There are lots of stem cells in adipose tissue, and they're finding that it's part of the endocrine system now. There's more about fat that they're studying, but you do go through hyperplasia, so you tend to add those cells. But you'll see here that there's the nucleus, and you have this fat reservoir, and that's what changes when you have too much energy coming in, so too much sugar, too much fat. These fat reservoirs get bigger, and so you end up with the hypertrophy, always the case, and then occasionally the hyperplasia. So you have different types of fat cells, and you have fat stored in two parts of the body. Right underneath the skin there's subcutaneous, and fat is a tissue, and the cells themselves are called adipocytes, and the tissue itself is called adipose. You have two major types. You have the white adipocytes, which is the abundant, that's like 80 to 90% of the body, and then you have the brown adipocyte, and what makes it brown, and then you have the in between, the beige, is you can see over here with the white adipocyte that it's got one primary fat globule and only a couple mitochondria, so it's not really meant for burning energy. It's really a storage component. It's like a big storage container. On the opposite end here, the brown adipocyte, you can see that it has all these little tiny fat globules, but it's dense with mitochondria, and that's what gets its brown aspect. And brown fat versus white fat is a metabolically active tissue. It's the tissue that generates heat. It generates particular hormone output. If you were going to store fat, you want to favor brown adipocytes. The white adipocytes, brown adipocytes are generally regional. So you have particular parts of the body that are brown and particular parts that are white. But for the most part, the white adipocytes are your storage, your brown adipocyte are your metabolic and your functional tissue, and then the beige is just like a transitional aspect in between. But these are your adipocytes. They're like balloons, and you can see the nucleus here. And this big orange-yellow globe, like the yolk of an egg, is all the excess fat storage. And those come together, and that's an electron microscope view of adipose tissue. You can see the blood vessels kind of coming through there. You see some of the supporting satellite cells. You see some of the extracellular matrix to kind of hold everything together. And each of these are round cells that are containing a fat lipid globule that has stored triglycerides when it needs it. And these are your fat storage. You can see the brown adipose tissue, like through the neck and through some of the, you tend to have more brown fatty acid or adipocytes when you're younger, and it converts more to white. Also, depending upon if you're under the influence of growth hormone, if you're working out intensely. But if you're a sedentary individual and it's just overconsumption, it's mostly white. You have two storage. You have the subcutaneous fat, which is right underneath the skin. And then you have visceral fat, which is the more complex, more health concern, which is underneath the abdominal wall in the abdominal cavity itself. So you can see that there's naturally some fat around the heart and around the organs, but if you start to get too much like intramuscular fat or intra-liver fat, so again, places where you store glycogen, if it's excessive energy over and over again and you have insulin resistance, you'll start to get fat impregnation in those areas and get fat where you're not supposed to. But this is where the fat is stored, and the next slide here we look at how the tissue looks. Here's your circulation, your blood vessel. These are your adipocytes. You can see that there's quite a bit of a stem cell matrix, and you have some macrophages here. And again, the adipose tissue itself is part of the endocrine system. These macrophages have different inflammatory or pro-inflammatory functions. And as we go from an unhealthy state or healthy state of lean to an unhealthy state of obese, we get changes in our tissue structure. And so you have adipokines, which are cytokines that are produced by the body fat, and those are part of our immune function and part of energy storage and so forth, and that regulates tissue remodeling, wound healing, and these are good things. In a lean state, these are typically anti-inflammatory in nature, and the hormones that are released here, like leptin and ghrelin, these help regulate our appetite so we're not hungry as much. As we start to get overconsumption and we start to get more accumulation of fat mass, our adipose tissue changes, and it changes from an anti-inflammatory environment to a pro-inflammatory environment. And some of that is because of some of the chemicals that are being stored in our fat itself, like the tissue itself becomes toxic. You get more turnover, cellular turnover, so you get cell death, and so you're getting more macrophage activity to go in there and clean those up. And a lot of those stem cells that were once dormant are now active, and you not only get hypertrophy of the adipocytes, but you also get hyperplasia. So not only are the cells getting bigger, but you're getting more of those cells that are turning into fat cells. The other way of looking at this too is that since lean adipose tissue is part of our immune function, and it's a pro-inflammatory or like an anti-inflammatory where it's considered a benefit, when you lose too much body fat, you also start to get declined immune function, and so you have increased risk of infection, injury, disease states. And so when we look at like the BMI graph, you look at when you go too far to the right, when you're obese, you increase morbidity. Also, when you go too far to the left, when you're too lean, you also increase morbidity. And that's why when you have like really aggressive bodybuilding type or aggressive training, you start to see massive changes in physiological structure, heat intolerance, and pain intolerance, and increased susceptibility to fractures and injuries and infections, because the body fat is a good thing. But the Goldilocks body fat, right? Just right, not too little, not too much. I include this slide here because we're going to talk a lot about it, and since we're talking about adipocytes, but one of the things to remember is that insulin is an anabolic hormone, and we'll talk more about it, but insulin is very anabolic to muscle tissue and to fat tissue. And in order for us to access this stored fat, right, if we want these adipocytes to go from hypertrophy to atrophy, we want those to reduce, and the way they're going to reduce is by them giving up, let's go to that slide. The way they're going to reduce is by them giving up this fat globule. This stuff in here has to come out and get into circulation so that it can be metabolized into ATP. But if you're in an environment where insulin levels are high through the blood, these channels are only one way. So if insulin is high, these receptors are going to be occupied by the insulin molecule. Your fat cells are in suction mode, not in ejection mode. And so when insulin is there and those receptors are open, they're intake, and there's no opportunity for outtake. And so the key to fat loss is controlling insulin. And this is just kind of the teaser that we'll get to in our later of our lectures. This kind of takes you through the metabolic pathways for when you're looking at the fat reduction, when you're looking at lipolysis. So you have your fat stored in the adipocytes, and you have your triglycerides as a storage form. You have hormones that have been released, so like growth hormone, cortisol, glucagon, and these hormones are going through the blood. These fats are going into the liver, and the glycerol is being converted to glucose. Fatty acids are being converted to ketones, or they're getting converted to other molecules where they can be transported through. Fatty acids can be directly oxidized through the Krebs cycle, through the oxidative system, and you get these partially metabolized fatty acids into ketones, and the ketones are used by the heart, the liver, the brain. Not the liver, but the brain. And it kind of shows that this whole thing that we talked about last unit about fat metabolism all needed to start with hormones here. And if you have insulin, this ain't happening. And if insulin is low, you still need to have the other hormones there that are going to trigger triglyceride release, which is called lipolysis. So that's looking at the fat metabolism. So the next thing we look at now is muscle changes, and we talked a lot about muscle hypertrophy and atrophy in the muscle function aspect, and we'll look more about it in adaptations. But when we look at the tiers of our muscle structure, right, we have the overall muscle itself, and as we would expect, if we start to increase our adaptations and stress of the tissue, we expect an increase in muscle size or thickness. So the diameter of those muscles are going to be bigger, the cross-sectional area. When we go down and we look at the individual fascicles, and each of those fascicles, we would expect to see those fibers increase in size, not in number, right? So we're going to see hypertrophy of the muscle fiber, but not hyperplasia of the muscle fiber. If we look inside the muscle fiber, however, at the myofilaments that are there, we will see an increase in myofilament density. And so we do see, because that's what's happening, right? You're getting more actin and myosin stored in that tissue so that you get better contractile elements, more cross bridges, and that's why you get stronger. One of the reasons why you get stronger. So hyperplasia is not a normal part of muscle hypertrophy or muscle growth. It's usually muscle hypertrophy, but it doesn't mean that we don't get the myofilament hyperplasia, where you get an increase in those that tissue structure. And again, here's your muscle fiber. You have multiple nuclei in the cell itself, and this is interesting because not only do you have nuclei here, but you have these satellite cells. And when you have an untrained muscle fiber, the very first time you get this hypertrophy, and you have these from that training, you get these satellite cells that fuse to the fiber domain, and you actually end up with more nuclei. So you don't necessarily have stem cells in the muscle fibers or in the muscle tissue, but you kind of have like what would be considered like stem nuclei. So you actually have more nuclei that are there. Again, in the nucleus, what it's doing is it's creating proteins, and what these nucleus are doing, or the nuclei, the plural, is they're producing the contractile elements, so you get more of this that's happening. So again, the takeaway is that muscle hypertrophy is the normal mode for this increased diameter. You're getting increased number you're getting increased fiber thickness, but not increased number of fibers. But you are getting more myofilament density, where your density in those myofibrils, because you're getting more myofilaments packed in there. Part of this muscle memory that is talked about a lot in the industry is usually related to the nervous system, but there is a part, because once you go through your first training or first couple trainings, you get that fusion of those satellite cells, so you have more nuclei to do the protein synthesis, that when you take time off from the gym and you get back into it, you can usually increase your contractile elements faster because you have more. Think of the nucleus as factories that produce the contractile elements. So since you have more factories that are in your muscle fiber than you did from before, you're going to produce those contractile elements much faster in the future when you do more training. So when we look at the endocrine system itself, these you look at the slide here and you can see the adaptations that we're looking for. Like I said, the amount or the synthesis of the hormone is a big aspect, but the time needed for clearance and for processing is one aspect. Where the future is for our research and what we're going to talk about is more in the receptors. So how can we influence the increase, the upregulation or downregulation of the receptors, and looking at how we interact or how the tissues interact with that. So this is where we're going to get most of our conversation, and where the future of supplementation and intervention and so forth is going to happen is in terms of not how do we produce more hormone, but how do we actually increase the sensitivity or resistance to that hormone. And again, testosterone is one of those things, as we talk about in our next lecture, where we look at sex and age-related differences, that testosterone is one of those things where when you look at, if you're to take a random blood sample of a man on the street, you look at a sedentary man and an active man, they're going to have kind of similar testosterone levels. In fact, it is actually a chance that the active man might have lower testosterone levels because of the amount of stress he's being exposed to. So it's not the testosterone itself that we want to increase, it's does the testosterone have a willing recipient? Does it have the receptors that are there? And it's actually exercise and training that the way you train and the things you do, that you can upregulate these receptors to make your body more sensitive to the hormone. Same thing with insulin and controlling body fat. You're not trying to suppress your insulin production. Well, you are a little bit by reducing your carbohydrate intake, but it's actually the high intensity exercise that resensitizes you to the insulin's receptors so that your body doesn't need to produce as much. So we're looking at this upregulation or downregulation, and this is kind of depicted here. When you look at the image here on the top, you're seeing this upregulation where you have hormone floating through the blood, and in order to make your body more sensitive to it so it has a stronger effect without changing the hormone, if the cell adds more receptors, you can get a bigger bang for your buck. You're getting more effect from very little hormone. When you get downregulation, it's because the body is saturated with the hormone and saying, hey, we have too much of an effect. You're too loud. We need to turn you down a little bit because I can't think. So it takes out some of those receptors to the point where you're here, and then sometimes you get stuck in this loop where the body won't bring those back on. So that's the overarching aspect of it, is that you manipulate your hormonal environment by the amount of intensity or rest or recovery you're doing. But what we really want to focus in is how are we taking care of our cellular environment, where we're trying to maximize our upregulation or downregulation of our receptors, making ourselves more immune or resistant to tissues, the things that are going to store fat and break down muscle, and making ourselves more sensitive to things that are going to burn fat and build muscle.